Biochemistry of proteins. Proteins are polypeptide structures consisting of one or more long chains of amino acids residues. They carry out a wide variety of organisms' functions, including DNA replications, transporting molecules, catalyzing metabolic reactions, and providing structural support of the cells. A protein can be identified based on each level of its structure. Every protein at least contains a primary, secondary, and tertiary structures. Only some proteins have a quaternary structure as well. The primary structure is comprised of a linear chain of amino acids. The secondary structure contains regions of amino acid chains that are stabilized by hydrogen bands from the polypeptide backbone. These hydrogen bands create alpha helix and beta plated sheets of the secondary structure. The three-dimensional shape of a protein, its tertiary structure, is determined by the interaction of side chains from the polypeptide backbone. The quaternary structure also influences the three-dimensional shape of the protein and is formed through the side chain interaction between two or more polypeptides. Each protein at least contains a primary, secondary, and tertiary structure. Only some proteins have a quaternary structure as well. Each amino acid consists of a carboxyl group, an amino group, and a side chain. Amino acids are linked together by joining the amino group of one amino acid with carboxyl group of the adjacent amino acid. Each amino acid's side chain has differing properties. Some side chains can be either acidic or basic, while others can be polar, uncharged, or non-polar. These characteristics provide insight into whether the protein generally functions better in acidic or basic environments, solubility in water or lipids, the temperature range for optimal protein functions, and which parts of the protein are found on the protein interior. Being in contact with the external aqueous environment, some amino acids contained within the polypeptide chain can even create ionic bonds and disulfide bridge. The location of certain amino acids in the primary structure dictates how the secondary, tertiary, and quaternary structure will look. The transcribed RNA contains multiple segments called exons and introns. Exons are segments containing the encoding sequence for protein synthesis, while introns are intervening segments of nucleotides that will be excised from the primary RNA transcript pre-mRNA in a process called splicing. Splice sequences are found at the 5 and 3 tails of introns, marked with the denucleotide GO at its 5 end and AG at the 3 end. Alternative splicing is a mechanism used to create different isoforms of the same protein so that several protein isoforms can be created from a limited repertoire of genes. Alternative splicing is now known to deeply influence biological behavior, sex determination, cell differentiation, and programmed cell death. The mechanism underlying alternative splicing is not fully understood yet, but histone modification signatures correlate with a splice site switching via chromatin binding proteins.